President Thomas Lingauer.
Good evening. It's so great to see all of you tonight here. This is a big crowd. Actually, I admit more than we expected even. So this is almost a record-breaking conference. I don't know the exact numbers, but we're very happy that we could get you all here. We're all together, and I promise you a super exciting conference, and we're going to start with a super exciting evening this evening. I welcome you all to Basel on behalf of the whole team, and I hope that you have a nice, cool time here. As you may know, the Society started with this conference. In 1993, we had the first small conference, ISMB, and the conference was so successful, and the area was blossoming so much, and one could also say exploding so much, that we founded a society about 5 years later in order to make that conference sustainable. And now both the conference and the society have become premier for the field globally. What are we doing? We are committed to the advancement of computational biology. We do meetings. We have a website that informs you about developments. We are supporting and carrying out training and education. And we're an advocate in the field, to the field in policy making and towards society. Our membership count has just hit an all-time high yesterday. We surpassed 3,500 members. And here you see the breakup. We're truly international. You can be a member. Many of you are a member, I assume. But you can do more. You can maximize your membership by becoming active, being a real activist. That's a minority of the members, and we wish there to be more. How can you become active? You can join one of our COSIs, of our communities of special interest. Those are the groups that support your field within computational biology. If you're a student, you can become part of the student council, very active part of our society. Or you can engage at the regional level in your own regional group. You can also help us carry out conferences, organize conferences and webinars. We're grateful for anybody who helps us actively in bringing the society and the field further. The ISMB Student Council is our jewel, I should say. It's the promise that this field will go into the next generations. These are young people that are excellent and active and committed, and we're so grateful for having this set of people that runs fantastic workshops and conferences and is active on so many levels. You can visit them in their booth here outside this hall. These are our COSIs. There are 20 COSIs. I like to compare them to the European states. We don't have quite as many COSIs as we have European states. You know, the number of states goes down, the number of COSIs goes up, so maybe at some point we meet. And we see ourselves very much also as a federation, where each COSI has its own individual character, but at the same time we have a joint coherence for bringing the field forward. If you want to hear more about what the society is all about and what it celebrates and what it criticizes and where it can be improved, please see us tomorrow around noon, 12:45, in the Singapore room for the town hall meeting. The more of you come, the more active we can be in responding to what you wish to see from the society. In the last year, we have professionalized the society regarding ethics. We have provided, developed and approved a code of conduct for members and published it on the website. And we also declared ISCB a safe space regarding ethics. and we wish and actually hold the members to adhering to this code. And today in the board meeting, we approved the installment of a committee that maintains our aspirations for the society adhering to these ethical guidelines. You can inform yourself about these guidelines on the website. If you're one of those who tweet, we encourage you to tweet and disseminate quickly what you learn in this conference via Twitter. And we're very grateful to our sponsors. We have had a very good sponsor turnout this year too. And you see all the sponsors here. There are sponsors at several levels, and you will see these slides as the conference goes on. This is the least we can do in order to give thanks to the people who donated money such that we can have a successful meeting. We have a special, for the first time also, a special initiative on childcare. Some of you delegates could bring their children, and the conference supports the caretaking of these children while you are attending the conference with extra money, partly from the society and partly from third parties. And here you see the sponsors that helped us set up this program for the first time this year. These are the two conference chairs. You will see more of them during the meeting. Nikki Mulder and Thorsten Schwede were in charge of preparing this conference, and let's give them a big hand right at the start. The professional way in which these conferences are prepared is always a source of admiration for me. Program overview, only very quickly. I will not say all these numbers, but we have many tracks and we have over 665 presentations. So you will have ample opportunity and choice and alternatives to seek out what you're interested in. Also a new feature in the conference are the CompBio Ignite talks, sometimes also called the unconference part of the conference. We're making an experiment here of improvising talks from students, 5-minute talks on their posters that can be proposed and asked for without any reviewing. So let's see how that works. So I'd like you to enjoy the conference. Again, welcome. And now we come to the first exciting item. I'm very happy to introduce the first speaker, the speaker of the opening keynote, Nikolaus Rajewsky. Nikolaus Rajewsky has studied theoretical physics and received a PhD from the University of Cologne with a thesis on theoretical statistical physics. He told me, I asked him for the title of his thesis, and I can't repeat it, unfortunately. I forgot it again, but it doesn't have anything to do with biology. I can promise you that. So after the first postdoc at Rutgers University, where he was still in the theoretical physics field, he then switched to biology on the occasion of a postdoc at Rockefeller University and had a subsequent assistant professorship at Courant Institute of New York University. And then 13 years ago, he came back to Germany as a full professor at Berlin's large and famed university clinic Charité, who you may know dates back to the days of Robert Koch and Rudolf Virchow. In 2008, he initiated and is directing now a new branch of the Max Delbrück Center, which is a national research center on molecular medicine in Berlin. And his branch is called the Berlin Institute for Medical Systems Biology, and since this is a mouthful, we call it BIMSB. So BIMSB is endowed with permanent funding of 18 million euros per year, which is also a bundle, and also has a brand new building, which could be housewarmed about 9 months ago, and right now has 17 research groups, most of them directed with people from abroad. Nikolaus' research area is systems biology of regulatory elements. He combines computational approaches with biochemistry and molecular biology for the purpose of studying the function and interactions of RNA. He has pioneered research on circular RNAs and their function, and with his latest research on tracking the development of single cells organism-wide, which we will also hear about tonight, he and his group were featured as one of the seminal contributors to what the journal Science has named the science breakthrough of the year 2018. Nikolaus is a member of numerous international advisory boards, such as for the Human Cell Atlas. Together with G. Al-Muzni from the Institute Curie, Nikolaus has coordinated the Lifetime Consortium, that's the name of the project, Lifetime, a pan-European consortium comprising about 90 research institutes and 80 companies. Lifetime wants to improve healthcare by mapping, understanding, and targeting cells during disease. To achieve this, Lifetime integrates and applies single-cell multiomics with machine learning and personalized disease models to human disease progressions. Lifetime was in the running for an EU flagship project, a funding format that endowed projects in the EU with 1 billion euros over 10 years. Unfortunately, a few weeks ago, the EU has decided to abort the whole funding scheme while the projects were in preparation. They, killed the scheme cold. But the Lifetime project has continued perspectives to be funded, so they're still talking to Lifetime of bringing Lifetime on stage. Nikolaus is an EMBO member, a member of the German Academy of Sciences Leopoldina, and has been awarded Germany's most prestigious research award, the Leibniz Prize, in 2012. He also holds a honorary PhD in human biology and medical genetics by La Sapienza University of Rome. I've had the pleasure of knowing Nikolaus for quite a number of years. We were partners in the large German research consortium DEEP on epigenetics. I've always been impressed with Nikolaus's intense curiosity. It's really something very special. I'm curious myself, but I don't rival his curiosity. And that's something that most people lose in childhood. He hasn't lost it. He's not looking for incremental research, but he has an uncanny way of asking the right, seemingly stupid questions that open entirely new alleys to research and lead to grand visions. And he has the drive and persistence for finding the answers. On all meetings where I had the pleasure of hearing him, he was the unspoken hero of the meeting. And I expect no less from his talk tonight. Nikolaus.
Thomas. Okay, clearly this is a setup, but an incredibly nice one. Thomas, thank you very much for this incredible introduction, and I want to thank ISCB for having me and giving me an opportunity to present the work that we do primarily in my lab. I think, yeah, there we go. Yeah. Okay, so let's start. I need to show you this picture. There was a long development behind this. This is BIMSB. This is the outpost of the Max Delbrück Center, now in the center of Berlin. So we are a few hundred meters away from the Charité, and this opens up for us now totally new avenues in collaborating with medical and clinical sciences, and we are very excited about this. And just very briefly, in BIMSB, we bring together compute and a wet lab at the same footing. Things are separated just by glass doors. And also lab space is shared between different groups, and the whole thing is meant to really promote collaboration and also visit. So if you want to visit, I think this is a place that will embrace you. And yes, so Thomas mentioned it, so we were, this physicist actually here in the middle opened our building. And we even explained single-cell sequencing, machine learning, and organoids to her, which she enjoyed very much and became a fan of Lifetime, something that I'll go into at the end of my talk. But it pays off. I think I learned at least what kind of a joy it is to have a scientist, a capable scientist, as a politician. And when talking to Bonnie, I realized suddenly with a lot of embarrassment that I'm not a member yet of ISCB. So I will apply right away after this meeting. But I just think that it's really important for us to not only do our science, but also try to communicate about it, to explain it to the public, and so on, in particular in the times that we have been exposed to recently. I don't need to further explain this to you, I guess. Okay, so Thomas also mentioned what was named Breakthrough of the Year, and I want to make very clear there have been contributions, key contributions from many labs to make this breakthrough happening. But our BIMSB is new. Group leaders are young, and so I'm really proud of this, that we have made a contribution. Now, what is really the breakthrough? The breakthrough is that if you assume this is a stem cell that will be differentiating on different fates, that we are now able to reconstruct this journey of these cells to their fate molecularly along the entire trajectory. And I think this is really a fundamental new way to tackle or to explore understanding how cells make decisions. And the reason is that I strongly believe that it is important to understand the differential between states, because I think it is really the differential that tells you about what kind of reasons are behind the differential, and really puts up the challenge, as Newton always put it, as a scientist to explain changes, okay? And I think to explain a change between a stem cell and a brain cell is very difficult, and it will always stay shallow. But I think that actually going and trying to explain, grasp these differentials is actually possible and meaningful. It's a little bit like what happened a few hundred years ago. I think that when the data became available to really quantify how planets move around stars, and then asking, well, okay, so what really is the reason behind small parts of these movements, differentials? So I think we are entering a time where this becomes possible, and I personally, I can't probably emphasize my excitement enough. Now, I mentioned single-cell sequencing. We started this very early on. We established DropSeq. I'll talk about this a little bit. But we put this together, or I should say this very talented student in the lab back then, an undergrad, put this together as a summer project for about 10,000 euros equipment cost, and it's very cheap. And so we were able to generate the first data relatively soon. And as you can see here, even this big physicist here enjoyed starting a single-cell run. Now, how does DropSeq work? This is not our method. This was published by the Broad, Markosko et al. Cell 2015. You have a piece of microfluidics, and you drive cells in suspension through it with some pumps. And from one side, you put in beads with a lysis buffer. Each bead has some oligos, 100 billion actually, attached to them that will act as primers to catch the RNA. There is some oil here, so droplets are formed, and if you do this right, you have beads and cells occasionally meeting in the same droplet. Then the lysis buffer will open up the cell, and the RNA can now be captured by the oligos on the beads. And then you collect this, amplify, and you have one library to sequence. And now this is actually, you can deconvolute this and quantify which molecules and how many of them were in each single cell. Why is it possible? The ingenious invention is in the oligos, which are attached to each bead. So they carry 2 barcodes. Each oligo has 2 barcodes in the nucleotide sequence. One barcode is specific to the bead. The other barcode is specific to the oligo. So this way, when you sequence later on RNA, and you will sequence always these 2 barcodes, and you can then collect all RNAs from the same bead or cell in one bucket. And you can actually amplify in whatever way you want, because you know the identity of single molecules. So you can get away of amplification problems and so on. What you're really doing with this, you count single molecules in single cells. Now, we optimize this, and so for hex cells, for example, we capture thousands of genes on average per cell. And we have developed protocols to fix cells so that you can do controlled experiments and collaborations and so on. Now I want to come to the first main topic, which is space. How is gene expression really organized in space? And probably one of the most famous examples is the fly embryo. Many decades ago, people discovered, and Nüssler and Vollert here in Germany, and Wieshaus in Princeton and others, that there are genes which are expressed in patterns, and if you perturb these genes, then there are developmental problems, and actually that the patterns in the fly embryo already tell you something about the part of the adult body that will come out. And you see here stainings for 2 genes in this beautiful blue stripe, one gene, and in red another gene, and they co-occur in the pink stripes. Now, what we did is with Rob Zinsen, who runs a fly lab in BIMSB, we collected embryos in a developmental window of about 10 minutes, so a precise stage, dissociated the animals or the embryos, and sequenced the cells that we got. This was great, but then we asked, okay, so but I mean, where do they actually come from? Because from the fly embryo, you really need to want to know where the cells were placed in space to actually interpret here the biology. And so we used an idea that was published by Rahul Satija and Detlef R. and John Marioni and others before, which is we took these known marker genes and then said, okay, so if I have a sequenced cell and I see the blue gene and I see the red gene in this cell, then I know that the cell must come from the pink locations, because these are the only locations where both genes are expressed together. So by using actually 86 or so markers in their combination, we were able to drill down the position of pretty much all cells to a reasonable degree to actually reconstruct the embryo on our computer. So we call this a virtual embryo. It has about, we know about 7,500 genes per cell. And so you can now do an artificial staining or in situ just on your computer with this. And we discovered some new biology, which I'll not go into. Okay, so this is exciting and tells you that we can reconstruct space. But I always thought that this is not really the right way, or is maybe only one way of addressing this problem. And I'll tell you the reason for this. Imagine these photos here, or parts of a photo, are your single cell sequencing data, okay? So one for each cell, and the colors shall stand for the collection of genes expressed in this cell. Now, what we want to do is we want to reconstruct the space where they come from, so we want to piece them somehow together, right? Now, let's simplify the problem and only look at one gene, okay? One gene. So we'll make this black and white. And now I'll do the dumbest thing possible. I'll just simply do a random configuration of these cells. So I put them like this, okay? Now, what you will probably realize is that this is most likely not the right way of doing this, because it doesn't look like an image. So what you can do now is, of course, go through all 5,000 or so configurations by which you can place them together, and you look at them and then you say, maybe I'll see the right one. And actually, very likely you will say, okay, so this is the right picture. One of my heroes here, actually. So if you have more genes, actually this becomes easier because the genes for each piece of the puzzle have to agree with the genes in the neighboring puzzle pieces. So the constraints become much more, and then you can even more easily and with less mistakes figure out that this is the correct solution. So in some way, the problem that I'm addressing is to solve a puzzle without knowing the image that the puzzle is about. And what I'm also doing kind of secretly here is that I'm assuming that in situ images, or the way the genes are expressed, is similar to these kinds of natural images from our environment or us. Now, probably the big differences, but we know that when you do in situs, for a whole number of genes, you will see nice patterns. That is usually actually the case. For many genes, you will also see salt and pepper, but there will be these genes which tell you about how the tissue is organized. The reason is that the physiology of a tissue is always connected to the genes in cells which make up the physiology. So there's some mapping here to be discovered. And the assumption that we're going to make is that there are some genes which are contiguous in space, okay? Not all of them, some can be abrupt, that's totally okay, but there are some genes which are kind of holding together parts of the tissue. So now that's nice, so we are thinking about a score, but the problem is to score all these combinations. If I have just 1,000 cells, which is not that much for a single cell sequencing experiment, I have, of course, to go through more combinations than there are stars in the universe. And so we didn't know what to do at this point, but then we had a very beautiful collaboration with the Friedman lab and actually Morny Zahn. Back then she was a postdoc in Nier's lab, but then became a Harvard fellow. And this is Nikos Karaiskos, a postdoc actually in my lab, who had done previously the fly assembly in the previous project. And so what Moore realized is, first of all, okay, so there are two spaces that we want to map to each other somehow. One is the expression space of single cells. We can build some kind of expression distance matrix between pairs of cells, for example. And then the physical space of the tissue. So you can do, for example, Euclidean distances, and you'll get a distance matrix between all cells. And now you want to somehow map that, okay? So in a way that cells which are near each other in expression space tend to be near also in physical space. And what turns out is that you can map this to optimal transport theory, and you can use linear programming to actually solve this. And so, to me, that was totally astonishing. And so the question is really probably how well does it work? So here is now reconstructing the fly embryo using really zero in situs. This is really now what the algorithm gives you. It knows, it doesn't know any marker. It's just trying to solve a very complicated puzzle without knowing the image. And as you can see, just for 4 random genes, it actually does pretty well. Here's a symmetry problem. The algorithm doesn't know what is up and what is down in the embryo, but that's actually not that bad. Now, we cheat a little bit, and we throw in 2 marker genes as information, just 2, and see if it improves. It does improve, actually, quite a lot. And you can show that when you're at 4 marker genes, actually the reconstruction is usually perfect. So what we learn is that this principle of the correspondence between physical and expression space seems to be strong enough to reconstruct actually a very complicated tissue from first principles. I want to stress one thing. This is a global optimization, right? So you look at the way of putting cells together that on a global scale, this kind of continuity optimization works. So there's no problem with local discontinuities or some genes being sharp, okay, and not expressed from one cell to the other, as you can see actually in these pictures. It is really about the global solution. Now, what we can do now is we can build algorithms. I'll not go into detail because I think that is a developing field and we are looking at some methods. I'm sure other people are looking at other methods to systematically discover spatial genes, genes which are somehow involved in organizing space. And so in the fly, I can tell you we can do this, and so we know a lot about the fly. So it's a very nice test case, and we find relatively easily genes which are spatial genes. But I now want to go to some other tissues, because you may say, okay, the fly embryo is very special. How can you reconstruct more differentiated tissues? And I want to give you some examples. This is the mouse intestine. It's an essentially one-dimensional problem, but it's a classic problem where the position within the tissue is essential for understanding the function. Because the stem cells sit here at the bottom of the crypt, and then cells march out, and it is very important. So they change a function while they are walking, okay? So it is very important to understand where the cells actually sit when you do single-cell sequencing, where you dissociate the intestine. So we ran the algorithm and it assembled the intestine. When we compared to recent work where people used many images to do the same problem, we got a very nice correlation. And then when we then asked, okay, can we discover novel genes which are spatially organized? We could do this because we can also compare. So this is a NovoSpark, our algorithm, to the known map or the reference map that was published. A similar situation is in mouse liver. This is, of course, not a one-dimensional tissue, but it is zonated, so you can walk in certain directions, and that is essentially one-dimensional again. And so, again, we were able to reconstruct this very nicely and also find new genes which are probably important for understanding how the liver is organized in space. Now I want to come to the probably most interesting, well, maybe the most interesting, but certainly the most difficult one in terms of applying these kinds of principles to, and this is the brain. So as everybody knows, the brain is comprised of a huge number of different kinds of cells with very different function, neurons, glia, and so on and so forth, many different neuron types. So can we also assemble brain structures? Now, very nicely, the Makosco lab has just very recently published a method to sequence RNA directly on sections of tissue. So we are looking now at a section of mouse cerebellum, and what you see here is on the computer reconstruction of one gene from a spatial RNA sequencing data from the Makosco lab. Okay, so at each point here with some resolution, they sequence the RNA directly on the tissue, and now we're looking at one gene. This gene is a marker for so-called Purkinje cells, so it has some nice pattern and marks you where the Purkinje cells sit. Now, if we now run NovoSpark and ask it to assemble the mouse brain, I'd expect it would maybe not to work so well. But you can already see that when just using 5 markers, obviously not the Purkinje cell marker, you start to reconstruct the correct pattern, and it gets better the more markers you use. This holds not only for Purkinje cells, but also for arbitrary other markers that we looked at. Now, we can also discover novel genes, which we think are spatially important with our data now. And so we think that it tells you that also in the brain, this idea of that there are genes which are organizing space and that you can actually discover these genes and you can use them to reconstruct space is very powerful. Now, these are tons of complicated data, right? So we can do slices and let's say do like, such as Makosko did direct sequencing of points on this slice. You can do slices throughout the brain, so you can do maybe, let's say, 100 slices, sequence the RNA on each slice, and what you really want to do is then build the three-dimensional complete map of a tissue, molecular map. So actually that you really can have in front of you the expression of all genes in each part, cell, even subcellular, we think is possible, of the tissue. And we think that this will probably replace a lot of the current kind of approximations that we do, for example, when we do histology in the clinics. Now, so these are massive data. How do we actually even just interrogate them or look at them? And so there's a group leader at BIMSB, Stefan Preibisch, and he has developed imaging software for his entire career. So the Fuji software that many people are using, actually, he is one of the developers of this. And so we realized that he can actually easily tweak his imaging software to actually work with sequencing data. Now each pixel has 20,000 values, and so this is kind of an interesting problem. But he has done this, and you can now browse and use all kinds of imaging tools to actually play with your data. This is just one gene in 2D, right? And also works in 3D. And of course, what we need now are tools like Google for tissues or stuff like this, that lets you compare parts of the substructure of a tissue to other parts. Let's say what is disease, what is healthy, does it look like other structures that I know of or other people have known of. So we think this is a really very broad avenue to develop machine learning or other kinds of computational tools. Yeah, so that's, I want to point out where are we with our clinical histology, pathology? What happens when you go to the doctor and they do a biopsy and look at this? It is still what was proposed in the 19th century. You do slices and then you do some, chemistry that you have found out somehow is telling you something about the disease. Okay. This is still what happens, and I think that it is time to improve this. We are now able to replace this by full molecular maps, okay, and really systematic approaches to link these data to diseases or to patients in a specific way. And so I think that is clearly room for a lot of improvement. Now, that is from the clinical side, but I think there's also now for the basic sciences, there's a lot to learn here. I want to remember, you probably know this, but the spatial environment is incredibly important. So, an extreme example is the tumor microenvironment, of course, so where the immune cells sit in relation to other cells is important for understanding what the tumor is doing. Actually, it can do totally different things depending on this, as we know from a lot of research. But I think what is also true is that we are more or less blind to capturing what I call tissue genetic interactions, okay? So we know that there is things like mechanotransduction. An extreme example, if you switch on an oncogene in a cell, we know this from work from, for example, Stefano Piccolo, that this oncogene is now active, okay, but the cell will not become a cancer cell if the stiffness of the tissue in which the cell is residing is low enough. So if the tissue is soft enough, this cell will not get sick. However, if the stiffness is higher, then this cell will become a cancer cell with all the consequences that we know of. So there are extremely important interactions like this. There are now some signaling pathways like Hippo, which are known to be important of this, but I'm pretty sure that there's much more to discover. And we are starting to become, I think in the future, we can systematically identify these kinds of interactions with the kind of methods that I try to explain, and insights into how space is organized. Okay, now this was space, and let's now turn to time. So there was work from the Linderson and Karchenko labs, which allows you to predict the future transcriptome of a single cell. And I want to explain very briefly how this works. When we sequence RNA from a single cell, we will see exon-exon junctions, which stand for the mature message of that gene, but we will occasionally also capture an intron-exon junction, which is somehow the precursor of this gene. Now, what you can derive this equation from first principles. It tells you that the change in message is proportional to the ratio of precursor to message minus some turnover rate, the half-life, the turnover rate of the message, the stability of the message. And the intuition to explain this is that if you see a lot of precursor, okay, in ratio to the mature message, you can assume that the cell just transcribed lots of this gene and there was not enough time for this to be processed. So your prediction will be that the message will go up in the future. And conversely, if the ratio is the other way around. Okay, so you can then write just as a super simple Taylor expansion, the message in the future as a current message plus the velocity of that message times time. You do this for each gene, and so you look then at all genes together and you compute the overall velocity of this cell in a gene expression space, which is roughly 20,000 dimensional. So what you get is an arrow which points in the direction in which the cell will move in the future, and the length of the arrow will tell you the speed by which the cell will be doing this. And so we used this in a developmental problem. So we had, in my lab, we have an immortal animal which is called planaria, and this animal is immortal because it constantly replaces old cells by new ones by differentiating them out from a pool of stem cells. So all the developmental pathways are constantly active in that animal, and so we figured that when we sequence all cells of this animal, we should be reconstruct actually, in principle, the entire developmental lineages of this animal. So how the animal is made from stem cell to all cells it has. This is actually a complex animal with a brain. It hunts for food. It actually eats meat. And these are the data. So each point here is a transcriptome of a cell projected by some method into 2 dimensions. And we validated all of this. I will not go into this, but all of these little clusters here are meaningful cell types of the animal. Okay? And then we computed the RNA velocity for each cell, and you can now see here that we see how the cells are moving, and they're actually moving as we thought. These are the stem cells radially out into their differentiated phase. So actually the RNA velocity tells you predicts the direction in which lineages are occurring. And so we then with Fabian Theis, who is an expert in this, in particular with Alex Wolf, who derived a beautiful algorithm called PEGA for deriving these kinds of lineage trees. That's a whole other topic. But we were able to reconstruct this tree for the entire animal, which we think explains a major part of how the lineage tree is actually how the animal is made. This, in maybe 10 years, maybe in 100 years, will also be available for us humans. Now, what you can do is you can cluster now on this tree, you can cluster genes, and you can actually now find out, because you have the entire information, what combinations of genes are probably important for different trajectories or decisions within this lineage. So you also get hypothesis of how things really work, not just a description of what is happening. Now I want to leave this for a moment and tell you something about the cell cycle, which is unpublished. So this started with the question, okay, we know how the cell cycle looks in textbooks. It's kind of a circle printed on a page. But how does it actually look like in 20,000 dimensional gene expression space? So you could imagine in this space that it is really some complicated trajectory that comes back, because it's a cycle, we know it must come back where it started, but it could have really very different shapes, and we don't really know how it looks like. And so here's now the answer, what we think is the answer, at least in the cells that we looked at. We looked in various proliferating cells, I should say. And this is an example here for HeLa cells, which we sequence single cell very deeply. So you see this is a cell line where cells grow asynchronously, so they're in different parts of the cell cycle. And if you sequence each cell and you do a fancy principal component analysis, you actually get a circle. So this is not some kind of nonlinear projection, this is a gene expression space. And so when you get this circle, and then what we did is we took markers for each part of the cell cycle and just colored the cells according to these markers, okay? So and out came yellow, red, blue, and so nice that they form territories, that's great, but then even nicer that they are exactly reflecting the right progression that we know from the cell cycle. So this speaks for that this is reasonable. You can also sort cells and look at the total number of molecules. Remember that we count molecules in single cells. And so what we see here is actually that the RNA contents of these cells goes up until M phase and then drops down. And the factor is actually more or less 2, which is something that people have actually argued for. You can read all kinds of papers about this, but we see that overall this really is the case. And you can, of course, also look at marker genes and verify that they behave as they should behave. We now computed velocity using this model where essentially only transcription is in the model, right? So the other things are just assumed to be constant or not existent. So it is really a model only about transcription. How much can this actually explain about this circle? And the answer is that it explains exactly that it is a circle because the arrows turn out to point tangential to the trajectory as they should. So what we learn is that the minimal changes in RNA velocities drives the cell cycle. Why am I saying minimal changes? I'm saying minimal because this is a circle, and we know that to make it around with these tangential arrows, it's the minimal change in velocities. Again, the point is here that this is just explained by transcription. I think that the length of the arrows is actually not that meaningful because I think there's a lot of other regulatory layers, obviously, we know that. And I think that influences how cells, if they stop or if they proceed or not and so on. But the direction is exactly right. So I think the transcription tells the cells during the cell cycle which direction to move. The speed of this is probably modulated in many other ways. What we also found is, and I cheated a little bit because I just presented you the 2 dimensions from the principal component. It turns out that there's actually a third component. In the first 2 components, we found more or less all the cell cycle genes. Okay, so the cell cycle is more or less completely explained, I think, by the circle. But there's a third component, and when you look at the genes which make this third component, you will find out, I'll show you this later, that they have something to do with environment. But we then decided to do an analysis where we now look at the circle tilted by 90 degrees. So we are looking in this plane now. And what you can see is that the RNA velocity tells you that at the end of M phase, there's a tilt upwards, okay? And we think this is our model, and I'm happy to also discuss this in much more length. There are all kinds of other data coming together, but the model is that we have currently is that the cell cycle is actually this circle, but that it's tilted and drifts in the spiraling movement, and that the genes which are behind this direction of the spiral are environmental genes or genes that you could probably link to epigenetics. And I think the explanation is that when you do this experiment, the environment changes. You know, the nutrition changes, the cells do this, do that, so they age. So you have to react to this, and I think this is what we see. Now, this is nice, and you can push this much further. But we were also interested in asking, can we actually go with RNA velocity where we assume that it's only transcription? Can we go further? Okay? Can we also actually measure splice rates? Can we measure the turnover rates and so on? And so there is a fantastic RNA biochemist at the ISMB. His name is Markus Landthaler. And he is labeling cells all the time for thiouridin. It's a metabolic labeling of RNA for all kinds of purposes. And we decided to try to put this into single cells, okay? So the way this works, you feed for thiouridin, which is an altered uridin, to cells. They take it up and they incorporated this special uridin into newly synthesized RNA. Now, there are some chemical tricks that you can do, developed by other labs, so the result is called SLAM-seq. When you sequence RNA, then the U's which were incorporated, which are these 4-thiouridin U's, they are read out by the sequencing as a C, not as a U. Okay? So you can discover them because you know the normal sequence of the transcriptome, so you can actually find exactly the messages which are newly synthesized. And so we collaborated on putting this into a single cell. So there are many people behind this listed on the last slide. But we decided to make a time experiment. So we simply let the cells grow, hex cells in this case, and label them from minute 0 with 4-thiouridin. So and now here in this plot, we quantified the T to C conversions, so the number of newly synthesized molecules in the end. And you can see that this actually goes up, this is log scale, so this goes up very nicely. This is some background that you always have. I should say that ways of label RNA in single cells, many labs are realizing that this is very interesting, and so there are now preprints also, so there are a whole number of other labs working on this. I just want to present where we are in our Landthaler and my lab collaboration. So what we realize is we can now write down some simple equations for this. We can look at the precursors. So the old precursors can only decay, they can only be spliced, but new labeled precursors are transcribed. You see, transcripts are so long and the 4-thiouridin is so massive that each newly synthesized RNA will be labeled. Okay? So transcription only speaks to the labeled precursors. For the messages, if old, if new, we can write down some simple equations. Actually, they can be solved exactly as just linear. And you get 4 equations with a few unknowns, and you can actually now fit these unknowns to the data. So what you get is for each gene, and during the cell cycle, you get transcription, splicing, and turnover rates. What we do here is we sort the cells for each time point into S phase, G1 phase, and so on. We can do this with our methods. And then we do these fits for alpha, beta, gamma, for transcription, for splicing, and so on. And what we see is, first of all, if you look at this, so we plotted here 57 example genes just to not overboard you with data for some hundreds of cells in each phase. What you can see is that in S phase, transcription is actually lower than in G1 phase. And that actually kind of makes sense because there's a DNA, there's a lot of synthesis going on, and it's difficult to transcribe this. What you also see is that splicing, the time for splicing, so the time needed for a precursor to be spliced, it comes out in the order of magnitude of minutes, which we know from the literature is correct. Okay, but the dynamic range is really much lower than for transcription, which is actually also what many people know in this field. And actually, velocity assumes that it is constant, and so that's a good approximation for G1 phase. For S phase, we actually see more variation, and we have to investigate this. And the half-lifetime of messages, we see that in S phase it goes up as if messages were stabilized. Which is probably also makes sense because you want to maintain a certain protein output and not just be victim of lowered transcription rate. So this is currently a work in progress, but I just want to illustrate that I think we will be able to actually come up with a gene-specific modeling from these single-cell approaches, okay, where we capture transcription, splicing, export, and probably later on there are people already working on this on translation, okay. So I think these are very exciting directions because it will allow us to really put together a quantitative picture of how cells move in gene expression space. And personally, my dream is that we push it to the degree where we can really predict what cells will be doing. Very importantly, there's epigenetics coming in the sense of which open promoters, enhancers are present in single cells. This also can be measured. There are a whole number of groups working on this. And so in the end, I think what we need is some learning approach to really map the cis sequences that inform the transcription factors, other factors to make decisions. We need to link those to the expression data. Now, this is a keynote, so I'm allowed to speak in this kind of long-term way that is probably totally wrong, but you put me up here, so I can do it. Okay, I have 5 minutes. I want to be, there should also be some questions. I want to show you what we are doing how we can use these methods to look at tumorigenesis in a way that I think is a little novel. This is a collaboration with Walter Birchmeier. He has a genetic mouse model for inducing a solid tumor in these animals. This is a salivary gland tumor, which is a tumor that kills many humans. So the idea was if you look at different time points of the animals having this cancer, 40, 90 days, to extract the tumors, to dissociate them into single cells, and then run our methods. And this dissociation part is actually really difficult, and I'm hoping very much that this will be outdated soon because we can do these super beautiful slices, right, and then just reassemble everything so we don't have to go through the artifacts and difficulties in dissociating complex tissues. But we were able to reconstruct, first of all, the healthy salivary gland. Okay, this is a mouse model, so we know the genetic background, we control it. This is a healthy salivary gland before the tumor comes. And so we have all these different cell types, and we can actually put them together in this nice model of the salivary gland with all the different functionalities encoded by these cell types. Now, doing the same clustering analysis with the solid tumor cells reveals that actually, look at this. So we colored parts of this map. If there are many cancer cells present, then it becomes red. It's a kind of a heat map, okay? And you can now see which cell types are actually affected by the cancer. You can see that some cell types, at least at this level, are not affected at all, it looks like, and others are heavily affected. And there's one type that is actually totally specific to the cancer. It doesn't exist in healthy salivary glands, and these are actually, by marker analysis, the famous cancer stem cells, which we detected here by a totally unbiased approach. Of course, we find new markers here, and I'm not going to go into this. This naturally gives you a lot of new markers for this kind of cancer and interesting targets. I want to say that we can do better than just looking at RNA. We can actually, this is a method developed by Marlon Stöckius at the New York Genome Center. You can sequence antibodies linked to oligos, so you sequence the linked oligos when you put them on the cells before you put them into DropSeq. So this is a proof of principle. These are fax data, classic fax data for some antibodies on cells that we know, and you sort them with a fax machine into different populations. When we link the same antibodies to oligos, we put them on the single cells, and we now sequence in the droplets the antibodies that were specifically bound. You can do without any of these assumptions that go into some of the gating. You can put them into even maybe better 3 populations. And actually, this assay you can do for hundreds of different antibodies in parallel. So with the fax, I mean, you can maybe do 5-dimensional, but then it becomes practically not possible. So when we do this on the cancer, on the solid tumor, we actually learn that the relationship between protein and message is often very good, but sometimes also it is not. And so you learn more details about the cell types that are present in your tumor. I should say that the RNA from this experiment again reproduced the map that we had established before, so that's a control. Now, what it allows you to do is look at the so-called microenvironment in much more detail. So from the immune cells, we can now see that there are different types of immune cells in there, and you can start to make more sense of this. Importantly, we can, I think, reconstruct part of tumorigenesis. So this is a diffusion map approach, and we found out that the basal tumor luminal cells in the middle, cancer stem cells, are probably a trajectory. It could also be the other way around. We don't know this, but we think that this is a progression by which tumorigenesis is happening. And you also learn something about which kind of biological pathways, in which order they occur. But I don't want to dwell on this because I think it is too specific for this talk. Now, in my last 5 minutes, I want to tell you about lifetime. So we can now, as I told you, molecularly follow cells in space and time. So the idea is now to do this on disease progressions. Okay? We want to know the trajectories of cells in our body during disease. And so this is what lifetime is about, and this consortium that Thomas was mentioning. And what we want to do is, in this consortium, we have this ambition, okay? But you must have an ambition, otherwise nothing will happen. We think that we can improve medicine and push it better into the future. And so the essential idea is this. If you go nowadays to the doctor, you probably have some symptoms, okay? And then there will be some analysis, and in the unfortunate case, you have cancer or Alzheimer or any of the really big killers that burden our society. The symptoms are heavy, suffering is great, therapy is very difficult, and the reason for this is that these cells are totally different from what they used to be, okay? So once upon a time, these cells marched off into some direction in gene expression space, and now they have become cancer cells, and it is very difficult to push them back to healthy. And so the idea is that with the new methods, we can actually try to arrive at this point where we capture cells where they are still relatively close to healthy, and then intercept, so to push them back to healthy. And we think for this to really work, we want to understand the molecular mechanisms or other mechanisms that are responsible for this, because if you understand this, you immediately have targets that you may want to start the interception on, because the cells have just responded to this mechanism. So knowledge of the mechanism really helps you. I think if you're a full-blown cancer cell, you can understand something about some of the mechanism, but to then assume that this will help you in making the cell healthy is, I think, far from realistic. In many cases, just we know that this is not true because these cells are overall so far removed from reality, from healthy. So this is the consortium that has come together, 90 research institutes. So this is an incomplete map of them, just showing that they are, that we are really a pan-European consortium, and we want to combine single-cell multi-omics, machine learning, and personalized organoid models or other disease models, including mice models, to disease progressions. We think this will generate a lot of innovation, because what we do is we unleash very new methods onto diseases. So we think we will discover new things rapidly. So that's the status quo. So there were 2 evaluation rounds. I think we were the best life science proposal. So I have my arguments, so you can. There are 6 proposals left. 2 of them are life science. Then, as Thomas said, the flagship program has been cancelled by the European Commission, but we received €1 million from the European Commission to build a roadmap for Europe, and they're actually really committed, I have to say, to fold us into the new emerging funding schemes. So I'm pretty certain that there will be funding. I don't know how much any of this, but I think this will have a very significant impact. So the big lifetime white paper will appear in Nature soon. So I think we have, as Europeans here, an incredible opportunity to say what we want, okay? To make a white paper that will really have an influence on how the European funding and science, life science scheme and organizations are moving forward. So I hope that you can share a little bit my enthusiasm and the enthusiasm of the entire consortium. And I want to thank my lab. I think it's clear that there are some principles emerging in space and in time. In time, I think it's a minimization of expression changes or acceleration as a principle that I find very interesting. And I think there's now a link of possibilities to really unravel tissue physiology and the molecular control. And these are the people who did all the work. I have here my collaborators, but I must end because Thomas wants a discussion, and he's very right about this.
Thanks a lot, Nikolaus. I raised the bar high, and you didn't disappoint me. Thanks for that. Questions? So I have a question. You should come up to the microphone. There are microphones in the alleys here. So you talked about space and you talked about time, but you didn't talk about space-time. I assume that that's at least a challenge for visualization. Is it a challenge for computation too, and for analysis?
Yeah. So of course, it is possible to do time courses. The problem is that in the time courses, whenever you do the analysis, you kill the tissue, right? So you need good systems to replace this with good samples that are meaningful. But I think that there are a whole number of these examples. So not all diseases will be amenable to this, and probably melanoma, for example, much more than other things. But you can also look at other reads out that are maybe more indirect, like blood. So what's shed into the blood is maybe also a good proxy. I think computationally, of course, this is the natural big goal, right? I talked about this separately, but yes, you have to put this also computationally together and not treat it as independent. That's totally true. Is that a good answer?
That's a good answer. I've seen...
Hello?
Yes.
Okay, so this question is about atlases. So you mentioned the planaria, and then you say, okay, for human mouse, maybe we are 5 years away. Because I'm really looking forward for projects like the Human Cell Atlas to deliver references that we can use to align or to identify cell types in a more automatic way and not so manual way. So I would like to see your opinion on how far are we for having this human mouse atlas or references based on your experience with planaria?
Yeah. So there is a Human Cell Atlas, which is a super important consortium trying to come up with a reference cell atlas for healthy humans. And that is a super complex project on its own. It will be the baseline for a lot of things that we plan to do regarding disease. When I said that we will eventually have the same thing as we have for planaria for humans, I didn't say 5 years. I think I said 10 years or maybe 50
years.
or 200 years. So, okay, the problem is you're not done with just recording cell types. The point about the planaria was that it's really the decisions, the developmental tree that is behind the understanding how the cells are derived, okay? And that is much more complicated than just saying, okay, so here's a map of the molecules in particular cell types. And I don't even know how to do these experiments because you cannot do experiments on humans, so you'll have to do this in organoids in ways that are still there to come. I think for some organs this will be possible, but to do the entire human is science fiction.
Question here.
Beautiful talk, beautiful work. You mentioned space and time, and you talked about the locations of cells. You can apply the same logic, of course, within the cells,
Yes.
which you haven't talked about. And
Yes.
specifically, when you talked about splicing, you had this nice result where you showed for RNA velocity, see the G and the S phases, the G1 and the S, and you said it's, your estimates for splicing rates for G1 were pretty stable, but for S, you get a lot of variance. And so one question would be, do you want to hypothesize about the reasons, mechanistic reasons for those? And if you would map them in space, coming back to my first comment, whether you would see the mechanisms relate to whether it's co-transcriptional, specific granules where that is occurring. Do you have some results for that?
Yeah. So thank you for the question. As I'm an RNA biologist, I'm actually super interested in exactly everything, more or less, that you just mentioned. And I think the next frontier in all of this is subcellular localization. The beads that you can use, you can easily work with 10 micrometer beads. That is subcellular for most cells. So you can actually start to really get a rough picture of the localizations within the cells. So I think that technically speaking, now again in this vision-like thing here, in my function as telling you something like this, I think this will come. We are working on this, many other labs are working on this, so we will be able to look at polarized cells such as neurons in I think, totally unprecedented ways. Now, regarding the specific data I showed, I didn't tell you that what we observe is that if we don't model intron retention, we are actually screwed. And so this is also other labs are now starting to report this. So this is a very important phenomena that kind of is now emerging in its true importance. So there are all kinds of precursors which are not processed or stored somehow in P-bodies, maybe even, or we don't really know. So there are all kinds of complications which are behind these data. And so I think, yes, so in the end you will have to use all this. But I'm excited about the first approximations which already give you a direction in which to further go.
There's another question over there.
That was a tour de force, thank you. All the way.
I don't know if a tour de force is something good or bad.
It's good.
I never know.
All the way back in the Drosophila portion, if the ancestor of the Drosophila was translationally symmetric across the segments, then you would expect the segment polarity to drive a lot of similarity. Now, does that turn out not to be the case, or is this an algorithmic thing where you're able nonetheless to reconstruct the overall structure of the embryo? Does this make sense?
I think the only assumption here is that the only assumption in this case is really that cells which are close to each other are generally more similar in transcriptional makeup than cells which are further apart. That's all.
So the front of each segment is not, in fact, similar to the front of every other segment in a generic sense?
No, that's not the case. So of the stripes, they have different identity, of course. So we recapture at this moment of development really the entire embryo. So each cell is different, actually. Each cell is different, and this is what you see in the data. I just showed in situs, right? So if you would then choose other in situs, you would see that the stripes are different for other genes. So it's really the combination of all this that gives unique identity to each position.
Thank you.
All right, thanks a lot. Let's give a big hand. I have some. Thanks, Nikolaus, for coming here, for telling us your story. We're very happy he's not here for very long, so if you want to pester him, you have to do it tonight, okay? Tomorrow there is a lifetime consortium in Barcelona, so we're not happy that he's leaving, but we're happy that he came, even though tomorrow there is this other event already. Thanks a lot. And we have a reception now.
Thank you, Thomas. Thanks, everybody.
Sehr schön, super.